Hi there. Welcome to this lecture. In this lecture, we are going to explore the Python libraries that we are going to use throughout our course for handling our most of the libraries. Not all of them, most of the important libraries that we are going to use. So I will try to cover all of them. If one or two are missed, some sorry about that. So first of all, mastering these biological the data is a really complex process. So we need a lot of libraries. So understanding these Python libraries is really important. So first of all, a little introduction about them. So we are going to discuss Pandas, Numpy library, SciPy library, BioPython, Seaborn, Python VCF library, Python Sam library, HTSeq library, DendroPY library, Python Mol library, which is also PMol, very important bioinformatics library for visualizing the protein structure. Sklearns, also their a little bit application, their benefits, and a little conclusion. So, introduction. Welcome to this lecture on Python libraries for data science, scientific computing, bioinformatics, data visualization, machine learning, and a lot of things. So, Python is a versatile programming language with an extensive collection of libraries that can be used for various purposes. In this lecture, we will introduce you some of the most popular Python libraries that is used in different fields of science and data analysis. We will discuss how these libraries can be used together to solve the real world problems and also how to use this in your bioinformatics as well. So Pandas is a very powerful data analysis library and tool that is allow users to manipulate and analyze large data set with ease. It provides a variety of functions for data cleaning, transformation, exploration, making it essential tool for the data analyst or any scientist. So one of the example how pandas can be used in real world scenarios is in finance. So investment firms use pandas to analyze financial data to make informed decision about investment. With pandas, they can quickly clean and transform large dataset and complex calculations such as calculating portfolio returns and risk, the measures of how much investment is going to cost and how much is the risk in investing that. Numpy is a library that is used for scientific computing, particularly in your area of linear algebra and numerical analysis. It provides an efficient way to work with arrays and matrices, which are essential data structure in scientific computing. One of the main advantages of using NumPy in real life is its ability to perform vectorized operation, which allows faster your computation compared to traditional looping method, where the loop that you make in your C++. So this makes it a popular choice for your data analysis, machine learning, and image processing. For example, NumPy can be used to manipulate and analyze large datasets, perform mathematical operations on arrays, and generate random number for simulation. Simulation which is used to count how many turns per second is happening in your operating system. So SciPy is a Python library used for scientific computing. It provides a variety of modules for optimization, integration, interpolation, linear algebra, single processing, and much more. One example of how SciPy can be used in the real world scenario is in image processing, how images are optimized. So the ND image module allows a manipulation of multiple dimension arrays, which can be used to filter and enhance images. You're seeing on my right corner, there is an image and it is enhanced, its light is enhanced by using the NumPy operating system. So another example in the physics simulation where the obedient module is ODENTD module is used to solve differential equations for life. So BioPython, very important for our course. The Python is a powerful Python library that is used in the bioinformatics field. So it provides a range of tools working with biological data, including your DNA, protein sequences, as well as the tools for analyzing and visualizing the data. From your BLAS to phylogenetics to machine learning to much more. One of the key features of BioPython is its ability and written in the common file formats like GenBank, FastI, BED, VCF, and much more. This makes it easy to work with the data with a variety of sources and also include number of algorithms for your sequence alignment, phylogenetic, your structural bioinformatics, and much more, making it a valuable resource for many different types of bioinformatics research. Seaborn. Seaborn is a Python library used for the data visualization that builds on the top of Matlab. If you know about Matlab, you should be familiar with this. It provides a high level of interface for creating informative and attractive statistical graph. Seaborn can be used to visualize relationship between variables in large dataset, create custom color palettes, and plot multiple distribution. One example of how Seaborn is working in the real world scenario is in the analysis of customer churn for a telecommunication company. By using the Seaborn to create visualization of customer demographics, usage patterns, its service plans, analysts can be identify the trends and patterns that may indicate which customers are likely to churn. So this information can be used to develop targeted retention strategies. Python VCF. So Python VCF is a Python library working with the variant calling format. So if you know bioinformatics or genomics and stuff like that, you know variants. There are single variants of many things, so you have to deal with it. So which are commonly used in your genomic research to store information about your genetic variations that come from families to families. So VCF provides a simple and efficient way to read, manipulate, and write VCF file using Python. One example of how this is working in our real life is identifying the genetic mutation that is associated with your particular disease. By comparing the DNA sequence of the individual with and without disease, researchers can identify the differences where the bases are changed that is responsible for your disease. So VCF can be used to extract this information from your VCF files and analyzing it in your Python environment. So Python Sam. So it is a Python library that allows you to easily work with your manipulation of SAM and BAM file. So what is a SAM and BAM file? SAM file is a single alignment, something like that, which I'm also going to cover in another video as well, which is the next video, I think. So which are commonly used in bioinformatics research, SAM and BAM files. So it provides a simple and intuitive interface for interacting and analyzing the data within these files. So one example of how SAM file is used, it is also being used in the gene expression analysis, your variant calling analysis, and identifying the genetic variation between different samples. Another example is in the RNA-seq, which is used into quantifying the gene expression level from your sequencing data. Overall, it is a powerful tool for anyone working with SAM and BAM file. Yes, BAM file is binary alignment manipulation file and SAM is secondary, I think, and something like that. HTSeq is a Python library used for high throughput sequencing data analysis. It provides tools for processing and analyzing large-scale genomic data such as RNA-seq, ChIP-seq, and HTSeq can be used to count the number of reads to align specific genome features such as genes or exons. This information can be used to identify your differentially expressed gene or to study alternative splicing event. HTSeq provide functionality for filtering and normalizing your read counts as well as visualizing the results of the analysis. So DendroPY is a Python library that provides tools for hierarchical clustering analysis. Hierarchical clustering is a method of clustering data points into groups based on their similarity. DendroPY allows users to visualize the result of hierarchical clustering in the form of dendrograms, which are tree-like diagrams that slowly show the relationship between clusters. DendroPY can be used for the variety of real-world scenarios such as analyzing your gene expression data, identifying the pattern of gene expression, and also associated with specific disease. In this context, dendrogram can be used to identify a group of genes that co-expressed and may play a role in your disease pathogenesis. So DendroPY can be used in ecology to analyze species abundance data and identify the pattern of community structure, just like this, which is showing in the figure. You are seeing the red color light of dendrograms and green, blue, then again red. And if you move on to another Python library, which is PyMol, is a powerful molecular visualization tool that we can use to that allows researchers to use and create high quality detailed images of complex biological structures like proteins. It is commonly used in the field of structure biology and biochemistry to visualize protein, nucleic acid, and macromolecules. With PyMol, user can manipulate and analyze molecular structures in real life, allowing for a deeper understanding of their function and interaction. Some example of how PyMol can be used include studying protein ligand interaction, designing new drug, and analyzing mutation in DNA sequence. Sklearn is a powerful Python library for your machine learning tasks, such as classification, regression, clustering, which we are also going to do in our course. So it provides a wide range of algorithm and tool to help data scientists and machine learning engineering engineers to build accurate models quickly and efficiently. One example of how Sklearn can be used in real-life world is in the field of healthcare. By analyzing the patient data with machine learning algorithms from Sklearn, doctor and researcher can identify the patterns and predict outcomes more accurately. This can lead to better diagnostics, treatment, and ultimately improve patient outcome. So, applications of Python library. One example of how Python libraries can be used together to solve the real world is in the field of bioinformatics. Python and VCF can be used to analyze your genetic data, while Dendro can be used to cluster these data. Additionally, SAM files can be used to manipulate, and HTSeq can be used for sequencing data analysis. By combining these libraries, researcher can gain insight into complex biological systems, like making the biomarkers discovery, identifying the core-relationship of genes. Another example of how Python libraries can be used is in machine learning. Sklearn can be used for the classification and regression, and Numpy can be used for numerical operations. And using the Seaborn, you can better understand the data and communicate their finding by data visualization. So using these library combinations, researcher can build powerful predictive model to make accurate prediction about real-world phenomena. Like we are going to do in this course, we are going to take a data set and then predict the outcome of your uh, breast cancer, which usually happen, and we are going to predict how much predictability is there and how is the outcome that cancer is going to cause. And the benefits, there are a lot of benefits. You can use it for your multidimensional areas, you can use it for machine learning, bioinformatics, scientific computing, data analysis, data visualization, and a lot of other stuff. So Python can be used in the large open source libraries and things like that. So there is a wealth of resources along documentation, tutorial, and a lot of help that you can get from Python community. Additionally, many of these library cross platform, meaning they can be used on different operating systems such as Windows, Mac, and Linux. Concluding all these Python libraries, you can just listen to what is uh, written on the slide, but I'm also going to tell you that Python libraries are really important. If you're a bioinformatics student, you have to deal with the Python libraries. For example, BioPython is an essential library for bioinformatics research, and Seaborn can be used for stunning data visualization. VCF and SAM files are important for analyzing the genetic variation, and just like that, you have to deal with gene expression data, so you have to use HTSeq. By using these Python library, researcher and data analyst, scientists can save time and effort in analyzing complex dataset, making it easier to draw meaningful conclusions from their research. Furthermore, these libraries are open source and free to use, making them accessible to anyone with the internet connection. Yeah. So we encourage you to use these libraries and see how they can benefit into your own research work. With Python and these libraries, these possibilities are endless. To you, that is the whole phenomena that we are going to use. So that is it for this lecture. So if you want to learn more about the Python libraries, it just follow along the course, and we'll see you in the next lecture.